And a reminder about the SNAP groups. I posted the SNAP schedule as one of the pages on Canvas. So if you go to the Pages tab, you should be able to find it. And if you go to 2 meetings, whether it's SNAP or one-on-one, any combo of the two, you'll get 2 bonus points on your first exam. But you have to go to 2 meetings. Okay. Any questions before we get started? Back into chemistry. Okay, I think I have everything set up on Zoom here. There we go. So we finished up carbohydrates. And as a reminder, what we're doing, we are talking about different biological molecules, basically. So we have 4 different categories, remember? We talked about carbohydrates. We still have to talk about lipids, proteins, nucleic acids. So we'll get through those today. Like I said, I cut some information out just so we can get through this and so you can focus on the most important aspects of these biological macromolecules. Okay, so without further ado, let's get into lipids. Lipids are the only category where there's not really, the only commonality they have is that they don't mix with water. So they're hydrophobic. They don't have a specific monomer. So with carbohydrates, we had monosaccharides. A bunch of monosaccharides makes up any carbohydrate. Basically with lipids, it just kind of varies. So we don't define a specific monomer. It's kind of a weird part about lipids. There are 5 primary types in humans. I'm going to go through each of these fairly quickly. Fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. An important aspect of lipids, a lot of times, this is like fats and oils. They get a bad rap, but they have a lot of energy in them. So they actually have more calories, which is just a form of energy, per gram than carbohydrates. So they can provide a lot of energy for cells and for the body overall. All right, so let's jump into each of these types just pretty briefly. I think like 1 slide per type. Fatty acids and the triglycerides are really closely related to each other, and I'll show you why in a minute. Fatty acids you can recognize. All of these are essentially just long chains of hydrocarbons. So we have a bunch of carbons with hydrogens coming off. That applies to most of the lipids. Fatty acids are no exception. So on one end of this long, let me pull you guys over here. On one end of this long chain of carbons, you have a methyl group. So CH3, that's one of those functional groups that you need to be familiar with. It doesn't really stand out because it just looks sort of like the rest of the chain. On the other end, this is a good identifier of fatty acids. You have a carboxyl group, which is C double bonded to O, and then an OH on the other side of the C. So that's a functional group that you need to be familiar with, carboxyl. So if you have a methyl on one end, a carboxyl on the other, and it's just a long chain of carbons, that's a fatty acid. There are essential and then non-essential fatty acids. I'm not going to ask you to know which ones they are, but essential fatty acids have to be eaten in our diet. We can't make them. All the others we can make from other molecules in our body. We cannot make the essential fatty acids. Okay. That's really all I'm going to talk about with fatty acids. They'll come into play with triglycerides on the next slide as well. But they can be on their own or they can be part of a triglyceride. So triglycerides are just basically 3 chains of fatty acids, which you can see here. So these are just 3 chains of fatty acids attached to a glycerol backbone. So for you guys on Zoom, each of these is a chain of fatty acids. Here we have our glycerol backbone, which is 3 carbons with hydrogens and oxygens coming off of them. Triglycerides are also called neutral fats. A A fatty acid on its own, so just one of those chains, is slightly acidic, hence the term fatty acid. When it bonds to a glycerol backbone, it becomes non-acidic or neutral, so that's why it's called a neutral fat. The primary function of triglycerides is energy storage. So they store a lot of energy. And if you've ever had your blood work done, triglycerides are one of the things they look for in your blood. So too many or too few is a bad thing, but hopefully your triglyceride levels are kind of right in that happy homeostatic area. Okay, so fatty acids and triglycerides, really similar because triglycerides are made up of fatty acids, in addition to that glycerol backbone. And I'm not going to talk about saturated versus unsaturated fats. Don't worry about that. Okay, on to phospholipids. When you think phospholipids, think cell membranes. These make up the vast majority of cell membranes. We'll get into that a little bit today. But they're really important in holding our cells together, basically. They're also kind of similar to triglycerides, except rather than having 3 fatty acid chains coming off of the glycerol backbone, there's only 2. So here you have a fatty acid chain. This is our glycerol backbone, 3 carbons. And then there's also the phosphate group, which makes it a phospholipid. So you have this phosphate group at the top above the glycerol backbone. So glycerol backbone, phosphate group, and then you have the 2 fatty acid chains coming off the bottom. And you'll often see it, see phospholipids drawn like you see over here, just a simplified version with a head and 2 tails. So this would be the phosphate, and then this would be the 2 fatty acid tails coming off of the bottom there. So the head and then 2 tails. And I'll talk about the hydrophilic, hydrophobic in the next chapter when we get into the beginning of the next lecture here. All right. Yeah, structural foundation of the plasma membrane of cells. So phospholipids, eicosanoids. These you may not have heard of. Eicosanoids are basically important signaling molecules. You can identify them because they are usually just one ring. So you'll see a ring of carbons and then a couple of hydrocarbon tails coming off. They have a total of 20 carbons. And they play, like I said, a really important role in signaling between cells. So cells are always talking to each other using these chemical signals. Eicosanoids are one of those signal molecules that they use. Eicosanoids play a role in a lot of different physiological aspects in our body. I've listed a few here. So inflammation. Inflammation is just when like you're injured and you're swelling or something. You have a sprained ankle. Swelling is inflammation. Fever is considered inflammation. It's just a way for your body to fight some kind of injury or infection. So they're really important in signaling different areas of your body to become inflamed, essentially. Blood clotting. We know blood clotting is pretty important if you get a cut. Hormone action. So hormones are also chemical signals, but they have to be told where to go, basically. So eicosanoids will help with that hormone signaling. Labor contractions are controlled partially by eicosanoids. And then your blood pressure, as far as the diameter of your blood vessels. So eicosanoids will increase or decrease the diameter of your blood vessels, which will increase or decrease your blood pressure. So they're important in a lot of different aspects. They look different from the fatty acids, the triglycerides, the phospholipids, because they have this one ring and then 2 tails coming off. No phosphate group. So there's 2 tails, but there's no phosphate group involved, like with phospholipids. And then next we have the final category, I think, of lipids that I'm going to go over, the steroids or the sterols. So those two names are interchangeable. These also have rings, kind of like eicosanoids, but typically there's 4 carbon rings. So here you can see 4 individual rings with 1 small tail coming off. That's going to be a steroid. Cholesterol is the most important steroid. Everyone has heard of cholesterol, and it's actually the steroid that all other steroids are made from. So it's the parent steroid. You have to have cholesterol in order to make all these other really important steroids in your system. Other steroids can include cortisol. If you've heard of the stress hormone, that's cortisol. So if you have a lot of stress, your body produces a lot of cortisol. It can be good in some situations, but long-term can cause some health issues. And then the sex hormones, progesterone, estrogen, and testosterone are all other examples of steroids. All right, so those are our steroids. You should be able to tell the difference between those different categories of lipids based on what the molecule looks like. So for instance, steroids have those 4 carbon rings with a little tail. Triglycerides have 3 fatty acid chains, that kind of stuff, as well as some of their functions in addition to the overall structure. Any questions about lipids? That was kind of a really quick crash course in the basic lipids of our body. I was going to go into good and bad cholesterol, but I don't really have time. If you're interested in that, you can always ask me after class or in office hours. Okay, on to proteins. So we've done 2 of the organic macromolecules, the carbohydrates and the lipids. We're moving on to the 3rd one, that's proteins. We're back to having a normal distinguishable monomer, unlike with lipids. So for proteins, our monomers are going to be amino acids. So a bunch of amino acids strung together makes a protein, more or less. There are 20 amino acids, and they make hundreds of proteins. So different combinations of these amino acids, depending on what sequence they're in within an amino acid chain, determines what protein it is. So there's only 20, but they can obviously be mixed up in a lot of different combinations. The general structure of this monomer, the amino acid, is shown over here. So you want to be familiar with these different aspects of an amino acid. So you have, first off, the amino group. So N plus 2 H's. We haven't really seen a whole lot of nitrogen yet. So amino group, hence the amino acid. The other side is carboxyl, that's acidic, so amino acid. So the carboxyl we already talked about, that's a C double bond O with an OH. Those were also seen in the fatty acids, right? So that was on one end of the fatty acid. You have an H off of one side of the carbon. And then there's this R side chain, which is a combination of a bunch of different atoms depending on the amino acid. So the 20 different amino acids are each going to have a unique side chain, a unique R side chain. We're not going to talk about the differences in the side chains, just know that the R side chain is the part that differs between the amino acids. So here for you guys on Zoom, this R side chain is what's different between the 20 amino acids. They always have the amino group, they always have the carboxyl group, and then that hydrogen off in this case, the top. The R side chain is the only difference. We haven't talked a lot about structure regarding like carbohydrates and lipids. A little bit, just the overall kind of general structure. Protein structure is really important in protein functioning. So there's 4 different levels of protein structure, essentially. You start out with just a chain, a simple chain of amino acids. And that chain of amino acids folds over on itself multiple different times, and then multiple amino acid chains can come together, and that's what forms the eventual protein. So there's lots of folding and then multiple amino acid chains coming together. So I want you to be familiar with each of the levels in general of protein structure here. Primary structure being that simple amino acid chain up here. Secondary structure, there's a couple different options. You don't need to know the names for those, but secondary structure is where there's bonding between the amino acids that aren't already bonded in the chain. So it starts to fold over on itself a little bit in secondary structure. Tertiary structure, there's further folding and coiling. So it's just becoming more and more folded on itself as you move along this trajectory of protein structure. So you've got primary, secondary, tertiary. That's still all just one amino acid chain. So primary to tertiary, you still just have one amino acid chain. It's just kind of folding over and over on itself. And then the quaternary structure down here, this is the final piece. This is where multiple amino acid chains come together to actually form the final protein. So that's why there's different colors down here. Those are individual amino acid chains that have formed their tertiary structure, and then they all come together to form the quaternary structure. So here you can see these different colors. So multiple, the polypeptide chains that are mentioned here, that just means many amino acids strung together. Okay. So with proteins, like I said, structure is really critical for proper function. If the structure, say all the amino acids are in the right order in the protein, but for some reason it didn't fold over correctly on itself, it's not going to be a functional protein. The folding matters a lot with proteins. You can also denature proteins by exposing them to, say, extreme heat or pH. This just means breaking down the shape. It doesn't necessarily break the bonds between the individual amino acids. It just unfolds the proteins, and then that makes them non-functional. So when you cook an egg, that basically denatures the proteins, and hence the egg is no longer functional. So denaturation, that's why our body wants to stay at a very specific temperature and a very specific pH. Otherwise, our proteins can become denatured, which will ruin everything. All right. I think that's all I'm going to cover about proteins. Those are the basics. Oh, no, sorry. I lied. We're going to talk about functions of proteins. So there's a lot of different functions of proteins in our body. I'm going to talk about 4 in particular that I want you to be familiar with. These will come up again as we get into the different body systems that we're going to be talking about. So structure is one really important aspect of proteins. Proteins can be really tough and strong. So those structural proteins can include proteins like keratin and collagen, which you may have heard of before. They're often included or are said to be included in beauty products. So a lot of like shampoos will say, oh, it has a lot of keratin. That's because keratin is a naturally occurring protein that basically toughens and strengthens hair, skin, and nails. So the idea, I guess, is adding more keratin into your hair to make it stronger. And then collagen is actually deeper in the skin and provides some deeper layer structure to our skin. So you can have collagen injected into your face, I guess. That's mostly where people do it. It seems weird to me, but people inject collagen into their face to get rid of wrinkles and add some more structure to their skin. But it is naturally occurring. It just kind of breaks down over time. That's why it's part of aging. Okay, and then we have communication. So proteins are really important when cells are communicating with each other. And in order to move a muscle, a bunch of cells need to communicate with each other. So your cells are constantly communicating with each other and sending messages. They're also receiving the messages. So I'll talk about the plasma membrane and the proteins that are embedded in the plasma membrane. A lot of those are basically taking signaling molecules. When they get attached to these plasma membrane proteins, that's going to tell the cell what to do just by attaching or receiving that chemical signal. So that's what I mean by receptors. So those are receptor proteins in the plasma membrane, and we'll talk about those in the next chapter. So that's 2 of the 4 functions I want you to be familiar with. And then there's also something else to do with membranes, membrane transport. So we'll talk a lot about the proteins in the cell membrane. Not only do they receive signals, they also allow molecules to pass across the cell membrane, which is honestly pretty difficult for a lot of molecules. So they govern what passes and help with that transport aspect. And then finally, enzymes are proteins. Anytime you see enzyme, think that's a protein. So all the enzymes in our bodies are proteins. They are speeding up chemical reactions. They're not making reactions happen that wouldn't normally happen. They're just making them happen faster and easier, is a good way to think about it. So if we look at these graphs here, on the left, this is a reaction without a catalyst or an enzyme. So you have this activation energy is the energy that's needed to get that reaction going. When you include an enzyme or a catalyst, it just decreases how much energy is needed to get it started. Good? All right, finally, our last category, nucleic acids. This is DNA and RNA. We do have monomers here as well. So the monomer of nucleic acids is the nucleotide. Nucleotides are kind of the most complicated of the monomers, I think, from a structural standpoint. They have 3 distinct components. One being the nitrogenous base. Don't worry too much about what that is. I'll show you an image of it. We'll talk about it a little bit more when we get into genetics and chapter 4, essentially. So 2 chapters away. There's always a monosaccharide, which is a carbohydrate. You know that monosaccharides are the monomers of carbohydrates. So you have a nitrogenous base, a monosaccharide, and then at least 1 phosphate group. And I'll show you what this looks like on the next slide. So I think most of you have heard of ATP, adenosine triphosphate. You want to know what ATP stands for. Adenosine triphosphate is a nucleotide. And it is the most important energy source for cells. So when you think ATP, think energy. That is what's providing energy for cells. It doesn't, it's not like a bunch of ATPs get strung together to make DNA. This is just kind of a unique situation where ATP is a nucleotide. It's kind of its own, it's a monomer, but it doesn't ever become a polymer, which is a little bit strange. But ATP is the best known nucleotide and just stays as a nucleotide. It doesn't go into making a long strand of ATPs or anything. So let's look at what that looks like. Here. On the right is another nucleotide. You can kind of ignore that. So this is cyclic AMP. That might come up later in the semester, but don't worry about it right now. On the left here is where we have ATP, the structure of ATP. So over here, this is ATP. The nitrogenous base, there's basically 4 different nitrogenous bases, 4 different options. We'll get into those when we talk about chapter 4. They can have 2 rings or just 1 ring, but nitrogenous base is one part of the nucleotide. Here we have our monosaccharide, which is ribose, so a very specific monosaccharide for sugar. And then ATP, because it's triphosphate, adenosine triphosphate, it has 3 phosphate groups coming off of it. That's what gives it its energy potential. So this is where all of the energy is stored in ATP. So those are the 3 parts of a nucleotide looking specifically at ATP. You want to be really familiar with ATP based on the structure and then its function. Just a little bit more about ATP here. It is the most important energy transfer molecule. It stores energy that it gains from other metabolic reactions. So when reactions release energy, ATP can, well, a form of ATP can grab onto it and basically hold onto it until it's needed for some other kind of work that the cell is doing. Like I said, the energy is stored in these bonds between the phosphate groups. So the phosphate groups are the really important part of the ATP molecule. If you chop off one of these phosphates, that's going to release a lot of energy. That allows the cell to do work. So that's where the energy comes in. If you take one of these phosphates off, that's going to release energy and allow the cells to do whatever it needs to do. Transport something across the membrane, make proteins, etc. And that's all I'm going to talk about with nucleic acids. We'll talk a little bit more about like DNA and RNA when we get into transcription and translation very briefly in chapter 4. But for now, those are the basics of all 4 organic macromolecules. So hopefully that gives you a good base for understanding them. Now we're actually going to do kind of a little ungraded mini quiz to see how much of that you guys actually kind of remember. So like I said, your exam is next Friday, so it's a week and a half away. If you'll grab just a piece of paper or part of a piece of paper, there's only 5 questions. You only need 5 lines, basically. I'm not going to collect this. It's not graded. I just want to give you guys an idea of, I want you guys to get an idea of how much of this you picked up on, knowing this is only 5 questions. It's only dealing with the organic molecules we just talked about. But there's a lot of details that get really confusing with these organic molecules, I think. So see if you can answer these questions. I will say these are all fill in the blank without a word bank. On the exam, there will be a word bank, so it'll be a little bit easier. But again, there's no pressure right now. So see how much of these you can answer, and we'll go over the answers afterwards. All right, ready? So no notes. All right. So number 1, triglycerides. A lot of this I just went over, so hopefully you took it in. Triglycerides consist of 3 fatty acid chains connected to a what? So 3 fatty acid chains, that's the easy part of a triglyceride. What is it? What are they connected to? Just write down what you think. If you don't know how to spell it, if you kind of know what it is, just write down as much as you can. Second one, what are the monomers of proteins? What are the individual subunits that make up a protein? Okay, number 3. This goes back to something we didn't go over today, so it might be a little tougher. What type of carbohydrate category does lactose belong to? So that's a carbohydrate I wanted you to know. Is it a mono, di, or polysaccharide? At least you have like a 33% chance of getting that one right. Okay. Number 4. Which of the four main categories of macromolecules contain phosphate groups? I'll give you a hint, there's two. So your options are carbohydrate, protein, lipid, nucleic acid. Those are the four main categories. Which two of those contain a phosphate group in the molecule? And then the final one. What type of macromolecule are enzymes? Literally just said this. So again, one of the four main categories: carbohydrates, lipids, proteins, nucleic acids. All right, so let's go through these and see who thinks they got the right answers. All right, so what are the three fatty acid chains in triglycerides? If someone said something on Zoom, I didn't hear you. I just turned up my volume. Anyone? Come on.
Glycerol?
Yeah, exactly. Yep, glycerol backbone. So glycerol backbone. I don't think I wrote this up in the previous class, but that doesn't work. Glycerol, look it up. I do not have a working marker, sorry. Glycerol, glycerol backbone. What are the monomers of proteins?
Yeah.
Amino acids, yep. Amino acids make up proteins. You want to know the monomers of, well, the three categories that at least have monomers. And this is a tougher one. Which type of carbohydrate is lactose, mono, di, or polysaccharide? Yes. It's a disaccharide, but close. Thank you for guessing. It is a disaccharide. So it's one of those three disaccharides I wanted you to know. It's a combination. There's one glucose and one galactose put together. That makes lactose. All right, so this one's kind of tough, but I like making connections between different things. So which of the two categories have phosphate groups involved? Lipids, so phospholipids specifically. And nucleic acids, yeah, exactly. So one type of lipid, phospholipids, but lipids is the correct answer. Lipids and nucleic acids both have phosphate groups. So if you see a phosphate group, you don't necessarily know exactly what macromolecule it is, but you can at least narrow it down to two of the four. And then what are enzymes? Enzymes are what category of macromolecule? Protein. Enzymes are proteins. So I just said that when you see enzymes, think protein. An enzyme is a protein. Okay, so hopefully that gives you some idea of how much you're picking up on these macromolecule topics. If you didn't know any of them, you might want to just make a mental note of that and really start committing some of this to memory. These are all very fair questions. Like I said, if it's a fill in the blank, you'll have a word bank at least, so it'll be a little easier, or it'll be multiple choice. But if you were really just totally at a loss for any of these answers, definitely start studying. The sooner the better. At least kind of reviewing, looking back at those slides will be helpful. All right, so that wraps up our chemistry chapter. Yay. No more chemistry. Well, we'll still be talking about chemistry here and there, but not just chemistry. We'll now be incorporating all of that chemistry knowledge into some more anatomy-based or physiology-based topics. All right. So I'm going to get started on cell structure here. Share. So this is chapter 3, and so the next outline is a combination of chapter 3 and 4. You can take that into account as far as detail goes. You don't have to do like a full-on outline for each of these chapters, but just you can cut them down a little bit, I guess. Like the length of each individual one can be a little bit shorter. All right. So that is due Monday. All right, cell structure. So this chapter is obviously all about cells, since we are made up of cells. Understanding from the cellular level, we're actually building up. So we're moving from molecules to cells now, so moving up the chain. The first part I'm going to go over deals a lot with the membrane, essentially, the plasma membrane. And then on Friday, you guys are actually going to, rather than me going through each organelle, like the nucleus, the mitochondria, the Golgi, all of that stuff, I'm going to have you guys basically teach it to each other in groups. So we're going to do an activity on Friday that'll take up, probably like 30 minutes of the class or so, where I'll group you up. You don't have to do anything in advance. You don't have to prepare. I'll give you the information, but just a heads up, I won't be talking quite as much on Friday, which will be nice. So for now, the cell structure chapter, I'm going to focus on plasma membrane stuff, and then get into some transport across the plasma membrane. That'll happen Friday, probably. All right. So the cell theory is a major theory within biology. The most important aspect, I would say, of the cell theory is that all organisms are made of cells, all living organisms. So anything that's alive is made of at least one cell. If it's not made of cells, we don't consider it alive. Closely related to that is this tenet of the cell theory, that the cell is the simplest unit of life. So you can't break anything down beyond the cellular level and consider it alive. So it's the simplest unit of life. All organisms are composed of cells. It can be one cell. It can be many cells. And then the final principle of this theory that I want you guys to be familiar with, there's additional ones beyond these, but these are the 3 I think you should be familiar with, is that cells make more cells. So cells reproduce themselves. And we'll talk about the cell cycle and mitosis pretty briefly in here, and look at how cells actually make more of themselves. You can see over here in this diagram on the right, there are nearly 200 types of cells in the human body. We're not going to learn each and every type of cell, so you don't have to learn all 200 types of cells. But I did want to point out the type of cell basically determines its function. So here we have, say in the middle, these are stellate or star-shaped cells. Those are nerve cells. So they're shaped in a very specific way to allow for transmission of electricity, basically, which is how the nervous system works. We have these squamous cells. So you'll learn some of these in the histology lab and the histology chapter in here. So some of these will look kind of familiar in a week or two. So the squamous cells are kind of flat. Those are sort of near, the surface of our skin or lining our organs. Columnar cells are column shaped. They're really good at excreting mucus. So they're found in areas that need to excrete a lot of mucus. So the shape of the cell, the type of the cell really determines its function. So like I said, we're going to go back to that theme over and over again. Structure determines function. This is another example of that. So that's the basics of the cell theory. What makes up a cell? In a very broad sense, we're going to get into the organelles, like I said, on Friday. But in a broad sense, you have the boundary of the cell, which is the plasma membrane. I'll cover most of the info about the plasma membrane. a little bit today, and then we'll finish it on Friday. Surrounds the cell, it's made of proteins and lipids. I'll talk about that in just a minute. And then you have the cytoplasm, which is kind of everything inside the cell. That includes the cytosol, or the intracellular fluid. You'll see it listed as. So you'll see this abbreviation a lot. You'll see ICF, that's the liquid that's in a cell. It also contains the organelles, which is like the nucleus, like I said, the nucleus, mitochondria, ribosomes, all those good things. And then this isn't really part of the cell, but it surrounds the cell, the extracellular fluid, or ECF. So that's outside the cell. That really comes into play when we talk about the different tissues, because there's fluid in between the cells in these tissues. For the most part in this chapter, we're going to focus on these first two bullet points, the cell membrane and the cytoplasm and the stuff within it. All right, just a couple more slides here talking about the details of the plasma membrane. Hopefully this is a review for a lot of people. Most of the plasma membrane is made of lipids. And I said specifically phospholipids, right? So 98% of the molecules that are in the plasma membrane are lipids. The other 2% are proteins. The proteins are really important, but they don't make up a lot of the molecules. They're big and they're heavy, but if you look at a cell membrane, most of it's phospholipid. So I'll talk about the proteins separately. First, I want to talk about the different types of lipids that are found. You have phospholipids, which I already mentioned, really important in the cell membrane. So if we look, you guys over here. If we look over here at this little drawing, this is our typical phospholipid drawing. You have a head and then the two fatty acid chains coming off. So the head is hydrophilic. That means it does not repel water. It mixes fine with water. So here we have the hydrophilic head, and then the tails are hydrophobic, which means they're scared of water. They repel water. So that makes this molecule what we call amphipathic. If you think amphi like amphibian, they live in the water and on the land. Amphipathic just means it has a hydrophilic and hydrophobic side. And they are arranged in a bilayer, which you can see over here. So the heads face out. So if this is our molecule, or our cell, sorry, our cell membrane, the heads are facing towards the outside of the cell and then towards the inside of the cell. So mixing with the fluid basically, because that's the hydrophilic sides. And then the tails are facing each other and they're hydrophobic. They don't really allow water to pass very easily. And they don't allow a lot of other substances to pass through easily. So that's where the proteins come in. Proteins will allow a lot of stuff to go in and out of the cell, whereas the phospholipid bilayer will not. Okay, so that's the phospholipids. There are two other kinds of lipids in the cell membrane. One being cholesterol. So we do need cholesterol. Cholesterol is really important in our diet, in large part because it helps solidify our cell membranes. If we don't have enough cholesterol, our cell membranes are all liquidy and fluidy, which isn't necessarily a good thing. So you see these little 4 ring chains here, that's the cholesterol molecules. They're sort of interspersed into the plasma membrane and they help keep the phospholipids in place. The phospholipids still move around a little bit, but the cholesterol really helps them stay a bit more stationary. And then finally, so that makes up 20%. So now we have, that's 95% of the lipids we've accounted for. The last little bit of lipids that are found in the plasma membrane are called glycolipids. They are lipids with a carbohydrate chain attached to them. So for you guys on Zoom, this glycolipid, that green antenna looking thing coming off, is the glyco part of the lipid. So up here, this is our carbohydrate chain coming off of a lipid. And you can think of them kind of as antenna. They're really important in cell-to-cell communication. So they stick these little carbohydrate chains out and can communicate with other cells through that carbohydrate chain. Then we'll get into proteins on Friday and the organelles. So we'll stop there for today. Right? Yeah, okay. So we're done.